In 1992, a woman named Diane sat across from her oncologist and heard the same 3 words for the 3rd time in her life. The cancer is back. 3rd recurrence. Ovarian cancer. The kind that does not respond to negotiation. Her doctor had one last option, a drug so new, so experimental, that producing a single dose required stripping the bark from 3 ancient Pacific Yew trees. 3 entire trees. Destroyed. For 1 dose. That drug was Taxol. Today it generates more than $6 billion in annual revenue and has saved hundreds of thousands of lives worldwide. But here is the part nobody talks about. Before Taxol could save those lives, scientists were forced into an impossible choice. Save the patients, or save the forests. And the Pacific Yew tree was about to pay a devastating price. This is the story of how bark from a tree that loggers once bulldozed as worthless became one of the most powerful cancer drugs ever created, and the environmental crisis that almost stopped it. Number 1. Diane's story stands at the center of the crisis. She isn't one patient, but a composite, a reflection of dozens of women facing the same impossible odds in 1992. 3rd recurrence, ovarian cancer. The end of the line for standard treatments. For these women, hope arrived in the form of compassionate use Taxol. The catch. Every dose meant stripping bark from ancient Pacific Yew trees, destroying what took centuries to grow. The choice was brutal. Sacrifice the forest, or let the disease win. Diane's case captures the raw urgency and the ethical weight of this moment. She represents the thousands who waited for a cure hidden in a tree no one thought mattered. Subscribe for more stories where science, nature, and human lives collide. In 2nd place, the landscape for recurrent ovarian cancer in the early 1990s was bleak. Standard chemotherapy hammered every fast-dividing cell, not just tumors, but hair follicles, the lining of the mouth, and the immune system. Hair loss, mouth sores, and relentless infections became the cost of survival. With each recurrence, the odds worsened. Salvage options barely existed. Most drugs worked by damaging DNA or blocking cell division, but cancer cells found ways to keep multiplying. Chemotherapy's collateral damage left patients weaker, while the disease pressed on. The need for a new approach was desperate, a treatment that could halt runaway cell division without destroying the rest of the body. Subscribe for more stories where science, nature, and human lives collide. Number 3 on our list, the story shifts to a narrow ridge in Washington state, August 1962. Arthur Barclay, a USDA botanist, hikes 7 miles north of Packwood into the Gifford-Pinchot National Forest. He collects bark, twigs, and leaves from a Pacific Yew, just another specimen among hundreds gathered for the National Cancer Institute's massive search for cancer-fighting plants. 35,000 species, one shot at hope. Barclay's field notes trace the journey, dried bark packed in a cotton sack, shipped across the country to the NCY's screening labs. Years pass. In 1967, Monroe Wall and Mansook Wani at Research Triangle Institute isolate a crystalline compound from that bark. 4 years later, the structure is published. Taxol, a molecule unlike any seen before. What began as a routine collection in a forgotten forest sets off a chain reaction, linking ancient trees to the front lines of cancer research. Number 4. Susan Band Horwitz's discovery. In 1979, rewrote the rules of cancer treatment. Taxol didn't just kill cells, it targeted the very machinery of division. In her lab, Horwitz watched as Taxol latched onto microtubules, the protein scaffolds that pull a cell apart during mitosis. Instead of breaking down, these microtubules froze in place. The cell's command to divide was locked mid-sentence. Cancer cells, desperate to multiply, became trapped. Unable to finish dividing, they triggered their own death, apoptosis. This was a revolution. Unlike old chemotherapy drugs that shattered DNA or poisoned every fast-growing cell, Taxol offered precision. It stopped cancer at the moment of birth, sparing much of the healthy tissue. Horwitz's work, published in Nature, revealed a new target, the mitotic spindle itself. Suddenly, the Pacific Yew was no longer just forest debris. Inside its bark, a molecule had changed the fate of thousands. Subscribe for more stories where science, nature, and human lives collide. Number 5. The crisis hits its breaking point in 1992. Every patient's dose demands the bark of 3 to 6 ancient Pacific yew trees. These trees grow just a fraction of a centimeter each year. To supply clinical trials, harvesters strip away 60,000 pounds of bark annually, a number that translates to tens of thousands of slow-growing trees lost. The math is unforgiving. One patient, several trees, centuries erased in a single season. The yew's home isn't just any forest. It's prime territory for the northern spotted owl, already fighting for survival. As bark disappears, so does critical habitat. The Pacific Yew Act passes Congress, halting unregulated harvests and forcing a reckoning between medicine and conservation. The stakes are immediate, the trade-off brutal. Save lives or save the forest. The world waits for a solution. Subscribe for more stories where science, nature, and human lives collide. Number 6. Pierre Potier's breakthrough. Change the fate of the yew. In 1994, working outside Paris, Potier unlocked a new path, extracting 10-deacetylbacatine-3, 10-DAB, from the needles of the European yew, a tree that could be trimmed rather than felled. With a few precise chemical steps, scientists could attach Taxol's crucial side chain, transforming 10-DAB into Paclitaxel. No more stripping bark. No more sacrificing ancient forests for every dose. Potier's patents, soon licensed to Bristol Myers Squibb, powered a rapid shift to semi-synthetic production. The supply crisis dissolved. For the first time, medicine and conservation could advance side by side. Subscribe for more stories where science, nature, and human lives collide. In December 1992, the FDA approved Taxol for metastatic ovarian cancer, marking a turning point in cancer treatment. By 1994, its label included breast cancer, expanding its reach against some of the most aggressive tumors. Through the late 1990s, Taxol and its generics reached hundreds of thousands each year, with about 300,000 Americans now receiving Paclitaxel-based chemotherapy annually. At its peak, Taxol brought in $6 billion a year, a remarkable sum for a drug once sourced from a disregarded tree. The story evolved in 2005 with Abraxane, the albumin-bound nanoparticle version that gained FDA approval for metastatic breast cancer. Abraxane changed the delivery game by binding Paclitaxel to human serum albumin, removing the need for harsh solvents like Cremophor-EL. This shift reduced severe allergic reactions and cut infusion times to 30 minutes, giving patients safer, faster care. The impact went beyond lives saved, setting a new standard for refining natural compounds in modern medicine. Today, Paclitaxel and Abraxane remain vital for treating ovarian, breast, lung, and pancreatic cancers. The numbers keep climbing, and the influence continues to grow. What started with a handful of bark samples now shapes the lives of hundreds of thousands every year. The Pacific Yew's molecule, nearly lost to neglect and over-harvesting, stands as proof that nature's overlooked species can still unlock tomorrow's cures. Across every breakthrough, one truth stands out. Nature hides cures in the least expected places. The next medical revolution could be rooted in a forgotten forest. Subscribe for more untold science.